Hi, my name is Leah McDonald and I'm in lab section 29 and I'm going to be walking you through DNA translation. So, after the DNA is replicated in the nucleus, DNA is transcribed into mRNA, and that happens in the nucleus. And then the mRNA moves through the nucleus into the cytoplasm, and that is where the translation can begin. So, you see we have our enzyme. It's the blue. The enzyme is made of two subunits. And then in the enzyme, the mRNA, which is shown in green, translation is going to happen from the 5' end to the 3' end. So what happens is that's the mRNA, and the mRNA has a codon. And that codon right now is, the start codon is AUG. So what happens next is tRNA, which is transfer RNA, is going to come in. And the tRNA is going to come into the P site, and it has an amino acid on it, and our creatine is an amino acid. So what happens next is the tRNA comes in. Now what has to happen though is this tRNA has to have an anticodon that matches the mRNA's AUG. So this first, which is the initiator tRNA, that is going to have the codon of UAC. So this one has UAC. And that is because it has to have the anticodon of the mRNA. So the whole point of this is to make a protein. So now what we have to do is we want to build our polypeptide chain of amino acids. So what happens next is another tRNA is going to come in with an amino acid. And all of our next tRNAs are going to come into the A site. The P site only comes in for your first tRNA. So what's going to happen is once the A comes in, it's going to start activating and it's going to start moving the process along. So this amino acid is going to jump to that to make a chain, and what's going to happen is the tRNA that's in the P site is going to move to the E site for exit, and this A is going to move to the P along with that. So then this tRNA doesn't have an amino acid, so it's going to leave the enzyme and go into the cytoplasm and try and go and find another amino acid, because more than one tRNA can have another amino acid after it leaves. So then the process just starts again. Another one's going to come in the A site with an amino acid. These are going to jump over here. The P is going to move to the E, and the A is going to move to the P. And then you're going to have your chain keep growing. We'll show it one more time with this one. That's going to move to there. Your E is going to exit. Your chain's going to move over. Your P is going to move to the E, and the A is going to move to the P along with the chain. Now what has to happen next is the tRNA has to have a stop codon. So once that stop codon comes into effect, it will stop the polypeptide chain. So the stop codons can be UAA, UAG, or UGA. And once the stop codon stops, all of it exits, it leaves, and the amino acid polypeptide chain is able to make a protein. And then this enzyme can be used over and over again, and it will just continue making more polypeptide chains.